Hello, everyone. Our lesson for today is about microbial cell shape and extracellular structures. In this lesson, you will learn the basic nature of microbial cells with a focus on prokaryotic cells. Their evolution from the last universal common ancestor will be discussed. Furthermore, this lesson also tackles the different properties, the morphology of bacterial cells, as well as the structure and functions of the possible components that can be found outside the cell wall of bacterial cells. At the end of this lesson, you will be able to define a cell, describe the properties of cellular life, describe the bacterial size and shape, identify the components found outside the bacterial cell wall, describe the structures and functions of the components found outside of the bacterial cell wall. Now, do microorganisms differ in their cell structure? So you'll be able to answer that as we end the lesson later. So you've already known about the cell because you we already discussed this one in our cell and molecular biology course. So we know that the cell is the unit structure of all living things, the basic and smallest unit of living matter that was first described by Robert Hooke from a thin slice of cork. And you already know about the cell theory, like just a brief review. So the cell theory states that all organisms are composed of one or more cells. Cells are the fundamental building blocks of life, and cells arise from pre-existing cells. So we know that the modern types of cells evolved from the last universal common ancestor. This is proposed by Wolfram Zillig. And the evolution of this basic type of cells followed this. Okay, I think I have already discussed many times already in our genetics, in our cell and molecular biology courses. So because of these events, the two basic types of cells has actually has arisen or have arise yeah. So the prokaryotic cell and the eukaryotic cell. We've talked a lot about eukaryotic cell during the cell and molecular biology courses. So this time we'll be focusing more on prokaryotic cells. So prokaryotic cells, their DNA is not enclosed within a membrane and is usually a singular circularly arranged chromosome. Although some bacteria such as Vibrio cholerae have two chromosomes and some bacteria have a linearly arranged chromosome, but these are very rare cases because majority of microbial cells have singular circularly arranged chromosomes. Now, the prokaryotic DNA is not associated with histones. So histones are special chromosomal proteins that are found in eukaryotes forming the nucleosomes. Other proteins are associated with the DNA. Now, prokaryotic cells lack membrane-enclosed organelles. Unlike the eukaryotic cell, membrane-bound ang nucleus, mitochondria, chloroplast, the other organelles. Their cell walls almost always contain the complex polysaccharide peptidoglycan. And they usually divide by binary fission. So during this process, the DNA is copied and the cell splits into two cells. Binary fission involves fewer structures and processes than eukaryotic cell division. Now, cells in general have properties that are common to all, and there are also properties that are only found in certain types of cells. So some of these properties that are common to all are compartmentalization and metabolism, growth, and evolution. So compartmentalization is important to have that environment inside the cell. Okay, so the inside the cell will be distinct from the external environment. So metabolism is happening inside that particular cell. Growth is also a characteristic of all type of cells. So it could be increase in the size of cells. So usually for eukaryotic cells, but for microorganisms, growth is actually increased or more expressed as increase in the number of cells. And evolution, all cells are capable of evolution because we have genetic material that dictates the future of our progenies. Now there are also properties that are only found in certain types of cells, like motility. No, not all are capable of movement. Differentiation, not all cells can differentiate into other type of cells unless it these are we know now that in production of induced pluripotent stem cell could make one differentiate into any types of cells. But not all in a conventional, in a natural way, not all are capable of differentiation. And also communication, not all are capable of producing substances that could harm. No, that is a way for the cells to communicate. They produce cells, and this will the cells have different response not to the target cell. Again, the properties that are just found in some type of cells are motility, differentiation, and communication. Bacteria come in great many sizes and several shapes. Most bacteria range from 0.2 to 2 micrometer in diameter and from 2 to 8 micrometer in length. There are few basic shapes. The spherical ones are called coccus, plural cocci, meaning the cells are like berries. No, they may be round or oval. There are also the rod shape. They are called bacillus. Plural is bacilli, meaning little stuffs. Okay? And the third type is the spiral. Cocci are usually round but can be oval. They could be elongated or flattened on one side. When cocci divide to produce, the cells can remain attached to one another. So cocci that remain in pairs are after dividing are called diplococci. Those that divide and remain attached in chain-like patterns are called streptococci. Those that divide in two planes and remain in groups of four are known as tetrads. And those that divide in three planes and remain attached in cube-like groups of eight are called sarcinae. And those that divide in multiple planes and form grape-like clusters, there isn't really a pattern for their shape they are making. So grape-like clusters of cells, so they are called staphylococci. Bacilli, on the other hand, they divide only across their short axis, so there are fewer groupings of bacilli if you compare that to that of cocci. Most bacilli appear as single rods called single bacilli. Diplobacilli, on the other hand, appears in pairs after division. And streptobacilli are those that are found in chairs. Others have tapered ends like cigars. Still, others have oval look, and some so much like cocci. They are called cocobacillus. Spiral have one or more twists. The answer, they are never straight. Bacteria that look like curved rods are called vibrios, and there are many found in the marine environment. Others called spirilla. They have helical shape, like a corkscrew, and fairly rigid bodies. Yet another group of spirals are helical and flexible, unlike the spirilla. They are called spirochetes. So unlike the spirilla, which has propeller-like external appendages called flagella to move, spirochetes move by means of eggshell filaments, which resemble flagella but are contained within a flexible external sheath. Other shapes evolved could be star shape, like that of the genus Stella. They could be rectangular or flat cells, like the halophilic archaea of the genus Haloarcola, and those that are triangular in shape. So what determines cell shape? So the shape of a bacterium is determined by heredity. Genetically, most bacteria are monomorphic. They maintain a single shape. However, a number of environmental conditions can alter the shape of bacteria. If the shape is altered, identification becomes difficult. Moreover, some bacteria such as Corynebacterium are genetically morphic, meaning they can have many shapes, no? Not just one. Now, the structures of a typical prokaryotic cell and the components of a typical prokaryotic cells are organized into the following. Those structures or components that are found external to the cell wall of bacteria, the cell wall itself, and the third are the structures that are found internal to the cell wall. Among the possible structures found external to the prokaryotic cell wall are the Glycocolics, Flagella, eggshell filaments, Fimbri, and pili. The substances secreted on the surface of the prokaryotes and secreted outside are called glycocolics, meaning sugar coat. It's a general term used for substances that surround cells. The bacterial glycocolic is a viscous, sticky, gelatinous polymer that is external to the cell wall, is composed of polysaccharide, polypeptide, or both. It is made inside the cell and is secreted to the cell surface. So glycocolics could be of two types, the capsule and the slime layer. If the substance is organized and is firmly attached to the cell wall, the glycocolics is described as a capsule, just like this in the picture. If the substance is unorganized and only loosely attached to the cell wall, then the glycocolics is described as slime layer. So you have here a typical structure of a Streptococcus pneumoniae cells, which are negatively stained. In negative staining, you stain the background so that the capsule appear like a halo around the cells. So these are the capsule, these clear zones in between the dye and the bacterial cells. So again, if we compare them, capsules are organized and firmly attached. Slime layer are loosely attached and are unorganized. Capsule in certain species are important in contributing to their virulence. So when we say virulence, we are referring to the degree to which a pathogen causes disease. Do not confuse yourself with the term pathogenic. Pathogenicity is the ability of the organism to cause a disease. Now, capsules often protect pathogenic bacteria from phagocytosis by the cells of the host. For example, Bacillus anthracis. It produces a capsule of diglutamic acid. It was believed that only those that capsule-bearing Bacillus anthracis are capable of producing a disease. Some are not. So it is believed that they are able to escape phagocytosis because they are protected by the capsule. On the other hand, another, just like we talked last time about biofilms, EPS, in marine organisms, in marine ecosystem, they're called TEP as well. Produces these extra polymeric substances that are capable of conducive environment for biofilm production. Okay, so it helps the cell in a biofilm attach to their target environment. So how does a biofilm is formed? So initially, a microorganism will attach to the surface, producing this EPS, and then secondary colonizers will continue until third types of if they don't inhibit each other's growth. Then another colony may be formed when some of the cells will be detached from the existing biofilm colony. Flagella is one important extracellular structure in prokaryotes. Now, these are long filamentous appendages that propel bacteria, meaning it helps them in their movement. Now, how will you describe a bacteria or a bacterial cell that lack flagella? So we call them astricuous. But if they have flagella, they may be peritrichous or polar. Peritrichous are those with a flagella distributed in the entire cells. Polar, only at one pole of the cell. If they are polar, flagella may be monotrichous, isa lang at one pole, or lophotrichous, you have a tuft, pong-pong of flagella coming from one pole, or amphitrichous, there are both flagellum, flagella at both poles of the cell. Axial filaments are found in spirochetes. These are for motility of the microorganism. Spirochetes move by means of axial filaments or the endoflagella. So bundles of fibrils that arise of the ends of the cell beneath an outer sheath and spiral around the cell have structures similar to flagella. The rotation of the filaments produces movement of the outer sheath of the panels of the first spirochetes on a spiral promotion. Okay, so they move by similar to the way a corkscrew moves through the cork. Corkscrew motion probably enables to move effectively through body fluids. Fimbri are hair-like appendages that are shorter, straighter, and thinner than flagella and are used for attachment and transfer of DNA rather than for motility. So these are found in gram-negative bacteria. So Fimbri, singular Fimbria, can occur the poles of the bacterial cell or can be eventually or evenly distributed over the entire surface of the cell. They have a tendency to adhere to each other and two surfaces. So they are like a morsiko, no, when you put it on sa inyong tela t-shirt, mamilit sila. So that's how Fimbri actually works for microorganisms. So, pili are usually singular pilus, usually small, longer than Fimbri, and there are a number only one of other of one or two per cell. They're involved in motility and DNA transfer. Also, for twitching mobility and letting mobility of bacteria. They're also involved in conjugation. Okay, so to summarize our lesson for today, now you must be able to answer these following review questions. What is a cell? What are the statements of the cell theory? What are the basic type of cells and how do they differ from each other? How did the present type of cells evolve for the less universal common ancestor? How are the different properties of cellular life? What are the different shapes of microbial cells? What are the different structures found inside a cell and cell wall of microbial cells? What are the structures of these, or what are the functions of these structures? So if you have questions regarding our lesson for today, feel free to contact me through this email, or you can directly send me a key in your questions. You can directly question me through the Facebook Messenger group chat. So thank you for your attention and see you again in our next lessons.